Well, I had a great time talking with colleagues since yesterday and today. I was really impressed by the breadth and depth of the science and learned a lot from everybody. So thank you so much for indulging me, and thank you, Arthur, for inviting me. And so I'd like to talk about two stories about Alzheimer's, actually. The first part is going to be some of our work to look at non-coding variants in this. Then we're going to talk about the data we have. I think many of you are interested to learn what are useful for research. But again, just a quick recap of what we know about Alzheimer's. It's most common cause of dementia, and it's almost exclusively happening in older people. Most of the Alzheimer's patients, they have late onset, which sometimes defined as 65 years old or older. And it's the most common cause, and has a second top most cause of death. And we're talking about at least 3 million people in U.S. now have 6 million, I think, Alzheimer's, and worldwide more than $50 million patients. We don't have effective treatments, and there's some new clinical trials that could slow down, can seem to stop the progression of disease, but it's still far away from actually, I think, having a minimal treatment. And we'll see how there, but that's already a major progress. Diagnosis is very difficult, usually involves extensive cognitive tests and image data. Now we have these new technologies using blood-based biomarker that seem to be very promising. So it's really changing how we're starting, but still have a long way to go. But the most important thing is actually the gold standard is still post-mortem autopsy. You put the brain tissues under a microscope and you stand it properly, you see these two kind of hallmarks, senile plaques, and neurofibrillary tangles, and we know what are their molecular compositions. One is amyloid, beta amyloids, and the other is the tau protein, and we know what genes they're caused. And so there's that's your connection between pathology and genetics. So one thing we want to do is understanding the genetic landscape of Alzheimer's. Is pretty heritable. It has a between 0.5 to 0.7 heritability in the literature. So, and we started looking at gene-wide associated studies in the, I would say, the mid to late 2000s. And the idea is very simple. You have enough cases, you have enough age-matched controls, you collect their DNA, and they're really robust technology, starting with SNP arrays, and now we're doing whole genome sequencing. And then we look at, for the whole genomic region, where are the places where you see a big difference between case control. That pretty much suggests there is something going there that increases the risk of Alzheimer's. But the most important thing is the sample. So we need to have enough sample size for enough statistical power. And this is where we started. There are many consortiums set up, and one is Alzheimer's Disease Genetics Consortium. I was really fortunate to be part of this from the beginning. And the PI is Jerry Schellenberg, who moved to Penn in 2009 to start this study, and work with lots of existing study as well as Alzheimer's Center, including Alzheimer's Center here at Michigan, which is a major contributor, and Dr. Paulson is here as the director. We just had a really good chat about genetics. And this is here. And the other thing is we have array data, we have whole genome data, and we have it from different populations. Diversity is a very, very important thing because for different populations, you probably have different variants. So we have these samples, and there's some other studies that we're trying to look at, and some of them could have dementia data. And then we started working internationally because you really need that to, and especially Alzheimer's, I don't think this is something that you can just do within a country. You really need international collaboration for all kinds of projects. And this was published in 2013, and it's considered classic. When people talk about the IGAP study for Alzheimer's, that's the first one. And one thing good about working with Europeans and collaborators, you get to go to Paris a lot. So that was wonderful. I have some really nice pictures. But this is a paper that identified 20 genome-wide significant loci. So their genome-wide significant, meaning their p-value passes multiple testing thresholds. So you know they have very high confidence. And it turns out they are reproduced in other studies over and over again. That's the gold standard for GWAS. And what you see here is, of course, ApoE. We know that many of you have heard about it. It has the strongest risk, genetic risk for late onset Alzheimer's, but there are other genes that show up as well. In convention, we name the locus by the nearest gene. That's just a convention. It doesn't mean, although most of the time is the case, that is the culprit. It could be a lot more complex than that. And then we continue bringing more samples. We improve the imputation and genotyping arrays. And this is a study that published 2019, and you see more variants showing up. We have 5 that's new. And of course, as you're going to do the effect size is going to lower, you need more samples. And this is the latest that's published by Bellingham. This is the one where we start bringing UK Biobank data. And this number increases to 75. So it really increases the sample size. And we have a newer paper that's under review. I don't remember the numbers, but now in the hundreds. These are ones that are associated with the risk of Alzheimer's, a case control study. And the other thing is we're working with European cohorts, so it's really just looking at European ancestry, right? That's the one still, like for most diseases, that's where we get the most sample size. Now, this is some major summaries, a brief summary of the table 1. You have some new loci in the closest genes. Actually, one thing that's very interesting is you start seeing some of these genes, granulins and taus and the TNEL106B. These are associated with frontotemporal dementia, which is another kind of neurodegeneration. They don't have plaques. So they're usually thinking of it's just different kind of disease. So when they show up, we're a little bit concerned. Is it because like misdiagnosis and so on? We really talked with the IGAP study investigator, and they assured us it actually showed up in multiple studies, not just UK Biobank, for example. So there's some robustness. And I think we need to revisit this. And one thing we now understand we know this, but we're really starting to think about the implication of this is most of the people with dementia actually have mixed pathology. Maybe this is a result of that, but we got to do more about this. And the other thing really important, again, Dr. Perelman talked about this, is the importance of genetics. It tells you about the pathway, it tells you about biology, because you know there's something causal here, all right? You take the genes, from these loci, you see you can do pathway analysis, and things start to emerge. For example, interaction with like lipid metabolism, and looking at immunity. It was a little bit controversial before we're doing this genetics, like if you see immune signal, it could be just the death of neurons, so it's not caused, it's the results. And this shows there is some causal component to this. So it makes sense to look at immunity when you're thinking about therapeutic strategies. And the other thing I just said is you need to start looking at other populations. So ADGC did a multi-ancestry analysis. And you can see some of these cases, that's LRC4C, another new locus when you combine signals from different populations. And it shows up in non-Hispanic whites and Hispanics, but you don't see variants in Africans. So it is a variant that's specific to these populations. And you start to see these. But well, for these other populations, we don't have enough sample size, not nearly as close as Europeans. So there's still more work need to be done. But I pointed out, I think new directions look at like how you have the right statistical method that can combine variants from different populations. Usually it could be different variants hitting the same gene, right? So that's another thing we're doing. And there are other kinds of things that people can do. So for example, you can look at age at onset. You can look at the decline of cognitive decline, the rate of cognitive decline. You can look at associated with, say, there's some endophenotypes, like the level of proteins in CSF, and so on. So we actually built a portal, and I would encourage you to have a look at this. Think of this as a database of all the table 1s in GWAS analysis. And there are detailed metadata, the cohorts are used, the analysis models, populations, and have a look. And the newer version we're working on is going to triple the publication. So you can use this to do landscape analysis and so on. But have a look. Yeah, it's called ADVP. So this is a timeline. It's actually created by Dr. Schellenberg. So I really like showing this. It really shows how the way we think about technology and think about genetics continue to evolve. At the beginning, when you find the APP and as well as the ApoE, as well as some of these earliest studies are actually from families. And then you start doing, there's this area of candidate genes. We didn't have enough power to do genome analysis. So we assume, well, here's 1,000 genes that it's probably important. Almost none of them were like replicated later. But it's really until when we start looking at the genome-wide association studies that you start to have high confidence, high quality reproducible finding. And now we're at a stage that we're starting to look at whole genome sequencing. We've been looking at that for a while, and there are new challenges. So that's where we are. We're going to find more variants. We're going to find variants from different populations. So, well, what are the challenges we have? We have a lot of signals. And with whole genome sequencing, you're going to start looking at rare variants. You're going to start looking at mutations, as well as insertion, deletion, structural variants, and so on. Different kind of things we're going to look at. We have better idea about pathways, but we probably need to do something a little bit deeper than just saying this is like immunity is involved, like something better. So their direction to do. Genetic effects are heterogeneous as well as people. You need to look at different population, different ancestries. And I think like how do we collect additional phenotypes, especially when you're thinking about Alzheimer's, it's a slow progressing disease. You need longitudinal studies, and that's hard to maintain. All these different challenges, but I think we're making a really good progress. If you think about the IGAP paper between now, it's less than 15 years. So what we need to do next, of course, how do we make sense of all these genetic findings? How do we turn these associations into biology? I'll talk a little bit about some of the things we're doing, but I think there are a lot of possibilities. So one of the problems we're working on is actually this issue. If you look at the GWAS analysis results and you see where are the variants, sometimes if it hits the coding region, you know what it does. It changes amino acids. Most of the time, actually for many of the common diseases, what you see is 90% or more are non-coding variants. They're outside the gene boundary. Some of them might be hitting introns, but many are actually flanking genes. So what do? Well, we think they're probably regulatory, right? That's a natural assumption. It changes the expression level, probably in the cell specific. It's also because that's the thing we know from our paradigm. So it doesn't mean that's the only way to do it. But if that's the case, what can we do? So the question is to find this. There's some SNPs, and we know they're hitting, say, regulatory elements like enhancers, and they might regulate the expression of genes with different cell types. So which one is actually agreeing with the GWAS signal you find? So there are all kinds of statistical models you want to do. You want to look at the linkage disequilibrium effects between nearby SNPs. You want to look at the expression profile, the associated signal to see if the functional data agrees with the GWAS data. And you have this idea that this is probably like, your mental model of this. There's some enhancer, the SNP affects that enhancer, which interacts with the promoter region. So that's how, at least we think, how gene expression is. So looking at chromatin conformation, chromatin interaction could be useful. How do we integrate all these data to translate GWAS signals to affected genes, pathways, and tissue context? So the workflow generally looks like this. First, you start with all the genome-wide associated signals. There could be tens of millions of that. Then you change your whole genome. You look at the ones that say are genome-wide significant, and then you try to figure out what are the independent signals. Sometimes a locus could have 2 or 3 different variants. They're all significant. They probably represent different haplotypes, and then you start to correlate that with functional omics data. You could look at regulatory element signals, enhancer markers. You could look at gene expression, QTLs, that says, here's a SNP that correlates with the expression level in a particular cell type. So you link all these data. And then you want to see if there are additional evidence, looking at, say, validation, or even do a functional validation. Say you want to do CRISPR to see if indeed that enhancer, if you delete it, it's going to change the expression in that particular cell. So we developed a pipeline. It's actually a while ago, it's in 2018, with the idea that you can take the GWAS data and compare with different kind of functional omics data, and in the sense that you also see if the gene expression pattern coincides with the GWAS signal, and they will show similar patterns. That's what I call colocalization. And then that will give you a hypothesis about what genes are affected. It's called Inferno, standing for inferring non-coding regular variants or something, but I like this acronym. The student actually came out with something that's really nice. And Spark means we actually later implemented for a Spark architecture to scale it up. But in order to do that, you also need to have a lot of functional data. They're harmonized, they have metadata. So we also built this platform called Filer. Now we're at version 2. So what it does is really simple. We go to different kind of functional omics data. Like you have ENCODE for different kind of cell lines, you have nice robot epigenetics for like tissues and so on. You take all these functional data, and then you harmonize and generate metadata with harmonized cell types and assays. So we build this resource, and not just Inferno, but if you have other algorithms that use this kind of function, you can actually, I will invite you to have a look at this resource because it's a lot of work to normalize these data, but it's ready. So that's what we built. And we also have a pipeline called HipFG that does these normalization. All these are published. And if you're interested, you want to talk about it, let me know. We can talk about how you can use these data. So we combine Phyla with the Inferno workflow, and we look at the Bellingers data, 2022, and you start with 19 million variants across the gene. You take the ones that are genome-wide significant, and you do LD pruning, and then you have to expand that because for any signal, any other SNPs that are in strong LD could be the culprits because they all will show the same association pattern. So that gives you a set of about 10,000 candidates to do this analysis, and running through the pipeline actually gives you 14 variants that seem to align with all the evidence. So we don't expect to find a lot because sometimes the functional data might not be right and all kinds of other issues. It's very, very stringent, but we want to find out some things that are promising. And you can report how many cell types you have a co-localization, what are the enhancer you find on these, and as well as you can look at the if there is any binding sites that you see that SNP actually perturbs the binding site affinity. So all these are good. And one thing that's actually, I think they're all worth investigating, and some of these haven't been reported before, but this one is actually we'll look at because it has the fewest signals, the fewer things we're gonna test. So let's start with that and see what's happening. So this is what the data looks like when you pull the relevant tracks. The green one is the top signal. Some people call it sentinel in this kind of jargon. That's where the strongest signal in GWAS is from Billingers. And here's a nearby SNP that has almost perfect correlation, but it has all these other evidence where you can see that it's actually hitting some of the enhancer that's in the tissue type, but not this one. And they are 10 kb apart. Yeah. And they're nearby genes. There's one, SE661G, that's the name of this. The other is EGFR, some gene that we're familiar with. And we actually started looking at eQTL data. That seems to suggest that SNP is regulating eQTL. And there are all kinds of other things. We want to look at different cell types to see their expression, to see if they have IPSs that we can do experiment. We can look at community confirmation to see if there's interaction between EGFR and the SNPs and so on. And then you can do CRISPR and so on. We have a collaborator who did a CRISPR experiments on the cell lines, and it indeed shows that if you edit out the regulatory elements, the EGFR expression changes. So it's really promising. We need to do more things, but it's a good start. And this is what you can find from the literature. Of course, it's well known that's a very important gene for cancer, but there's some literature evidence showing it probably has some important roles in Alzheimer's. And the more important thing is there are a lot of drugs that target EGFR. So it could be a way to think about drug repurposing. So this is one way you can use it. And if you do it for like genome-wide, then another thing you can do is maybe try to find out the biology. If you look at one locus, there's some ambiguity. It could be like multiple SNPs and they all regulate slightly different genes in different kinds of cell types. What we hope is when you look at dozens of them, they probably point to the same kind of cell types. So we came up with another algorithm called BTS. And well, when we came up with that, it's called Bayesian Tissue Score, but I don't know how many of you also know that BTS stands for something else. Yeah, that's the generation gap, sorry. So BTS is a boy band from Korea. And it's real story, actually. There's another colleague, and who's at Case Western, he published a paper called Pokemon. I was like, how you can do this? Okay, my next tool is going to be called BTS. It just happens. It has a really, really nice name for it. It's called Bayesian Tissue Score. But we have some kind of Bayesian framework in the idea. So we're doing the Inferno pipeline, but we have a probabilistic setting that allows you to calculate. And the probability setting is actually based on another classic paper called Penter. that does this kind of fine mapping things, like we have signal, which one is more causal. But the people in lab actually spend a lot of time refining the statistical model and figure out a faster way to compute it, as well as there's some algorithmic engineering effort to, so you don't need to recompute statistical alternative use it. So what this does is you can look at the genome-wide association, do all the QTL things we just showed, the integrated, and look at across all the different tissue types. That's where Filer becomes really, really useful. And then you can report a genome-wide enrichment score. So this is one thing, and we need to look at the Alzheimer's data grid. But one part of Alzheimer's, we're not exactly sure what's the right cell type. But there are other diseases we have a pretty good idea. It's like we look at irritable bowel disease. So we see immune-related signals showing, but also GI tissue. So that's the kind of thing. And here's an example of looking at coronary artery disease. And you see, here's the loci, here are the different kind of functional data. You see strong enrichment. They're all in blood vessels. That's a good sign. And there are different kind of regulatory elements. Some are on the histone markers, and some are on the DNAs. That's chromatin accessibility. We usually implies regulatory functions. So, well, the next thing we want to do is try it on other GWAS to understand this method better, refine this. But this is also an example where I truly believe it. If you look at all these genetic signals and see what's happened, it will give you some new insight about biology. Okay, so one other thing is actually we're pretty proud about this. The algorithm is much faster. We do a lot of engineering. That's why you can do it for many, many diseases and so on. And that's really important. So I'm going to talk more about this project called ADSP, Alzheimer's Disease Sequencing Project. And it's a major strategic effort funded by National Aging to do large-scale sequencing. One of the projects in the consortium is to do all the things we just talked about, generating expression QTO, find out what SNPs are regulatory in what tissues, and link all these data to refine the GWAS data. And it's a much bigger effort. They gather lots of QTL data with different kind of omics. They have the expression bulk sequencing, single nucleotide, single cell analysis, protein, splicing, methylation. So all these data, they're running QTLs on 13,000 samples on genome. So it's a lot of collaboration, and we actually, it's a group effort that we need to get the data, clean it up, and run all these analysis. And it's about 4 years of effort, but we're close to getting it done. And this is, I know this is the latest numbers, but we talk about 6 molecular QTL types, and they're reporting more than 250 million significant associations. So that's the first step. These will be used to refine, do the fine mapping of GWAS to find out the tissue context. We built a portal. It's not ready, so I'm not showing the URLs, but it will be soon. And it shows all the signals. Well, it's a little bit more than what we just saw, so that's great. And you can actually go in to look at the SNPs, look at the what are the QTLs associated, as well as looking at the genes and find what associated. And we also integrate that with the genome browser, so you can look at all these signals. Yeah. So anyway, so that is what we've been doing. I think it's pretty interesting. I hope this leads to a new biological insights. But most importantly, I think if it tells us about the right cellular context, I think it's going to be very valuable. So for the second part, I'm going to talk about some of the data and infrastructure we spent a lot of time building. And I hope that some of you will find it useful. If you're interested in understanding the data, and feel free to send me an email anytime. I'd be glad to talk with you about it. I want to start with talking about this repository called NIGATS, which stands for NIA, National Aging Genetics of Alzheimer's Disease Data Storage Site. So NIGATS, we started building this in 2012 and turned out to be the really important infrastructure for ADSP and other genetics data. It's highly secure. It follows the NIH genomic data sharing policy as well as the latest security requirements. And it's entirely developed on Amazon Cloud. So that's the only way that we can have people around the world to access. And the other thing is, if you're familiar with dbGaP, you apply data, then it's exactly the same process. so you can apply for access to the data. So it started in 2012, and we started building this data sharing service in 2018. And now we have, this is a little bit outdated data. We have 145 data sets with different kind of modalities, and it's 183,000 subjects, 230,000 samples. We have whole genome, whole exome sequencing. We have SNP arrays. Some of the functional data is there, but it's not that much. There are other repositories that focus on this. And there's some major data sets. We host ADSP, this whole genome sequencing. We also host the SNP array data from Alzheimer's disease research centers, and they're all harmonized with proper imputation, so you can download them and just readily use. We have 15 batches, probably about 20 to 30,000 genomes. And the other thing is we host a genetic data for a health and retirement study, and that's an NI-funded study. It's actually a headquarters here at University of Michigan, so we work with them. We host their SNP array data, and down the road, I think they're doing whole genome data. And then we can have all these data combined together and can do analysis with a bigger control. And the other thing we do is we make sure these data are interoperable with other resources, with samples, biosamples, NACRA, that's where all the samples are hosted. We can find out what samples are available for the data we have. With the clinical coordinate centers like NAC, that takes all the clinical data from Alzheimer's centers, Alzheimer's patients. And AD Knowledge Portal is dedicated to multi-omics data for a few cohorts, but with very deep data. Their genomes are actually in ADSP. And then there's a link with imaging data. They have the raw image data, but we have some process data there. So ADSP, that is the biggest data. And we now have, it's a group effort. We have more than 300 investigators, even more research staff that are working together since 2012 to put the whole study together. And our goal is to understand the genetic architecture of Alzheimer's. And it's a challenging problem, but I think we're making good progress. And we set up a pipeline to make sure all the data are of high quality. We try to minimize the batch effect, starting to enrolling participants, working with the cohort, so we make sure we can share the data legally, satisfies all the policy requirements. They're sequenced by different sequence centers. And then all the data come to our site at Penn. Then we process and consider with the same data, so you don't have batch effects, which is gonna cause problem. And then we have a way to share all these data. So you come in, you apply one data access request, you can access all these data. We actually got data from many different sequence centers, like 1, 2, 3. There are 10 different sequence centers, but they're all using Illumina short-read sequencing, and that's a good thing, actually. It turns out data we sequenced long ago with shorter reads, and now it's much longer with different, they're consistent enough, actually. We can combine these in a journal. It doesn't cause serious batch effects. And these are like some of the different studies, and they're pretty consistent. People are really good at this. So we don't need to redo the sequencing data for samples a long time ago. Well, people are starting to look at long read sequencing, so things might be different. Some samples will be resequenced, but this is an incredibly useful resource that you can cross-compare with clinical data and so on. Currently, we have 58,000 whole genomes from 57 cohorts, and the next round is going to be 74,000 genomes. Hoping we can finish processing sometime in the summer. Sorry, there's no data. The other thing I do want to point out is one thing we're really proud of about ADSP is it's a really diverse data set. About half of them are non-Hispanic whites, European ancestry, but the other are non-European ancestry. And this is, I think, pretty unique and rare in different kind of data. So we're really proud of it, and it's really important for understanding the genetics of Alzheimer's. We're also looking at not just like your typical SNPs, we're looking at indels and structural variants. This is actually Arthur is part of the ADSP structural variant work group, looking at different kind of things. I think Arthur's looking at transposer elements, but there are people who are using different colors for structural variants, copy number variations, and so on. And here's one paper that's published by one of our investigators, Wan Ping Li, who find a new risk variants associated with structural variants. And I think that's really promising, and there's still a lot more things that we got to look at. The other thing ADSP is really adding value is we're harmonizing phenotypic data from different cohorts. And we have a PhD phenotype harmonization consortium, and we just released the latest release last week. So now it's at release 4. What they do is actually they would take new cohorts, and they have a statistical model to be able to compare, to reprocess the data so they're comparable across different cohorts. For example, cognitive tests. Different studies will have different, cognitive tests they're using, but how do we generate an aggregate score that actually is meaningful across? And that's what we're doing. And the other thing is the image data. You want to have the same process like pipelines to turn it into some meaningful data, say the volume of the region of interest. So it's comparable across different image, equipments, platforms, and sequences, and so on, different resolutions. So this is a breakdown of the data. I think it's really incredibly impressive, and most of these are new. ADSP releases data before we even have a chance to do analysis ourselves. So these represent new opportunities. If you have some interesting statistical models you want to try, well, here's the opportunity that you might find something that nobody has found before. So we talk about the PhD, that's one of the additional consortiums set up. We have a group that does functional genomics. They're the ones that are running the XQTO project. And then we have a group of people who are using AI and machine learning on these data, especially integrating different kind of omics and image data. The effort is enormous, and there's still cohorts that are contributing data. We have worked with 86 cohorts, and some of these are actually outside the United States. That's where you can get more diverse samples, and that's a major effort. And of course, one of the issues we know is we need more non-European samples. And here's one project I'd like to just mention in case that something you're interested. We have a study called Asian Cohort Alzheimer's Disease. We're focusing on Asian Americans. There are studies that have Asians from Asia, but here we're focusing on this minority population. And there are many challenges for recruiting, retaining, and doing data collection, and so on. But we done our best to overcome many of these implementation issues. And our goal is to recruit 5,000 subjects in 5 years. And if you do you do Alzheimer's using say the NAC protocols, many standard protocols, you'll find the data collection, which is a little bit blurry. So I'm sorry about that. We're using a lot of established protocols, so the data can be comparable. And at this moment, we have 5,000 individuals that are on our interest list, and we have enrolled about 2,000. So I think we're a little bit behind, but I'm pretty optimistic that we're going to reach a really good number and make it one of the biggest studies for targeting Asian Americans for dementia research. And eventually, we have snippet rates, we have blood-based biomarkers. All these data will be available to researchers. I want to talk about another part, like all the data we just mentioned before you need to apply for access. We have many data like Filer, I just mentioned. You don't need to apply. You can actually just go there and just download or use what we call open access. And we talk about ADVP. We talk about Filer. The other thing I want to talk about is the Genomics DB of Alzheimer's. It's something that I think some of you find useful. Well, if you're familiar with, say, a Nomad database or UCSC genome browser, this is a little bit like that. It's a fully featured genome database with a visualization tool, but it's focusing on Alzheimer's. So this is where you find Alzheimer's GWAS signal or other things, and with complete annotation. What does this SNP do? What does genome from it? It has really nice features. You can start from a GWAS summary statistics, genome-wide, and you can look at all the top variants. You can search your favorite SNPs, and it's going to tell you some information. It's a breakdown of what data it come from, and you can look at the zoom in, just look at a signal like region, and it generates detailed annotation. So it has a lot of things, and there's the other thing we're working on. I hope that some of you will find it useful. Is we have a API that you can write programs for programming query. And it includes all the different databases we're talking, including one other thing I didn't mention is variance for quality of the variants in it. So any ADSP data you do analysis, you find a variant, you'll find all the records in these data. And it's using standard formats, like the things you look familiar as these are using the JSON format. These are things that you can write web URL queries and get it. It's very, very flexible. And then you can view all these things in an integrated genome browser kind of visualization. You can see that this is a case where you're taking genome-wide summary statistics and overlay with other functional omics data annotation. And we're still working on that. There are additional visualization technology we want to try, but they all come from these data you can run queries. So let me summarize the kind of data in ADSP. I think it's a really valuable resource, and some of you will find it interesting. I'd like to see if we can help you access that. We have 58,000 genomes. Some of the samples also have the earlier exome analysis, and they come up with some basic covariates, but we're adding more deep phenotypes. That includes neuropathology, cognitive test results, image data, CSF, like cerebrospinal fluids, biomarkers, and so on. And they come with all these genomic annotations, GWAS summary statistics, variant annotations, and all these are interoperable. They have the same genome builds and they have the same symbols. So it will make you, especially if you're doing, say, AI, machine learning analysis, I think this is a valuable resource. So, well, one thing we need to do is to just sell it to our funding agents, like how much impact, how many students. So it's a little bit hard for repository because one thing I learned is people don't necessarily cite the repository in their paper, but we do our best. So actually, some people in my lab did this, and I think that's another challenge for the GWAS study because people tend to want to use all the data in one big paper. So the number of papers is going to be a little bit lower because of that reason, but we have 422 papers that actually acknowledge our data explicitly, and they're cited by more than 12,000 papers, about 28,000 times. But I think what's important is like, after you have these genetic insights, what's the consequence? Does it lead to additional things? So look at more citations cited more than 500,000 times. And many of these are highly cited papers with clinical variation. PubMed has a way to assess that, and it continues to grow. And the other thing is these results are used by researchers worldwide. So I think that, and this number is going to continue to go as we get more data. Actually, this is something I'd like to see if anybody has given us, what's the best way to measure this metric, like impact, right? But it will also help us understand how to like prioritize our features and how to serve our community better. So some of the other things I try to share with you are thinking about, it's about the omics data we gather. It's about how we can link the genetic data with other functional omics and image data. And there's some barriers. The first is certainly identify what data is available and how to gather them. How do you make them available to the researchers, and how do you help them make sure that they have the right expertise, they have the right resources to use this data, their publications, and presenting the findings. If they have summary statistics, how do they return to us, and providing the results back to the research community. And of course, outreach, education, help other people understand what's the best way to use these resources. So there are many different barriers that we're facing as supporting a project, but also using these data. So NIGA is trying to do some of these things. In particular, how do we help everybody to do collaboration? I think it's really important. NIGAS can play a more active role than just like telling you, here's a website, do it. I think this is like, I had a great time talking to everybody. If there are places, there are occasions where I can talk with you, help you understand what data you have, and help you address some of the challenges, and help you understand the cohort and so on. I think that's a, that's a great way to work together to advance science. So I really look forward to talking with everybody. Feel free to send me an email anytime. And we're also thinking about, well, we have more data, they're more complicated. We're working to set up a cloud analysis common, so you don't need to download the data, but can analyze these data on the cloud. And we want to do more interoperability, so we can find and link data from different repositories, because that's how NIH is going to go forward to different repositories with different kind of data. And I think that's also another thing that this is heading. We need to do more interdisciplinary research. We need to know how to talk with each other. Say one thing I really enjoy being a bioinformatician is I can talk with methodology people who develop algorithms and tools, but also people who understand biology, who want to answer biology questions. We all need to talk with each other, not just like take the data and do some analysis and publish, but truly understand what's the biology question. So it's going to be about synthesis. It's going to be about looking at all the genetic signals and see what that tells you about the biology and all these things. And I do think that, for all of us who are working in the field of computational medicine, for bioinformatics, we're going to play some important roles to bring people together. Okay, so, well, thank you for your time. And of course, we have a lot of people to thank about generating all these data, and including our program officers, our advisors, and here we have both Mike Benke and Hank Paulson here. They've been giving us advice about the project over the years. I really want to thank them for their contribution. And thank you so much for your time. I'd be glad to take some questions. Any questions?
Thank you for your nice talk. I have a question. How do you address the privacy concern of patients? Because, for example, the nationality is very specific, that we can identify who the patient is, things like that. So how do you address that, like cryptography or things like that?
Yes. Well, the first is all the clinical data are de-identified according to, say, HIPAA rules. And we don't hold EHR at this moment. So that part, I think we're pretty good. The challenge is going to be genomic data, but in some sense, it's considered kind of identifiable, but only if you already have the person's genetic data and you do matching. So it's a little bit different from like when we usually talk about identifiable data, because you can't just use that data alone to identify people, right? So in that case, we want to make sure that people who use our data, they guarantee they agree with the terms of protecting data, and not just the person who applied for data, but also the institution, because they need to sign out the request. Say, if you are applying for our data, it's University of Michigan that signed off and make sure it's all. And NIH has been escalating the security for genomic data. And some of you know the new control access data repository, CADR, is giving us a lot of headache. But I think that needs to be done. And I think data access to, especially patients' data, is both a right for researchers to some extent, but it's a privilege, because every time people contribute data, they want to be useful. They also are taking the risk of, like we point out, privacy identification. So we gotta be really, really careful, but in a way that doesn't make it too difficult for research use. So that's the balance we're gonna strike. Yes.
Wonderful database, wonderful results. So there are some reports of variants that are like protective in the context of another variant. So Christchurch in the context of ApoE4, and a sort of second twist to this is, I don't know why nobody cites this, but there's a paper that sees a very strong effect of a mitochondrial, not uncommon variant that's highly protective against ApoE4.
Yeah.
Do people even look at mitochondria in GWAS? And so did they just never see it? And B, do you have any way of looking at interaction in your database?
Yes. We don't have data that you can directly look at interactions in that. But if there are like studies that do this analysis and report these results, we can do it. That's actually a very, very good idea. Mitochondria, yeah, there's some people in ADSP who are actually calling variants in mitochondria. It's a little bit like a copy number kind of things, because you look at those as, yes, we have that kind of data, and people are starting to look at it. Yep.
Thanks for the nice talk. I just have a question. So since this is largely Alzheimer's disease focused data portals or data pools. I wonder what kind of participant characteristics. Do you only recruit patients with certain definitive diagnosis of Alzheimer's disease, or you also recruit patients with a mild cognitive impairment? And also in terms of diagnosis, do you also include other different aspects of follow-up plan, because this is ultimately is a progressive disease?
Yes, we have these. The first is ADSP, and there's other GWAS data as well, so ADGC data. It's a contribution from thousands of cohorts, and every cohort has their own recruitment criterion. They have their own diagnosis criterion, and we do have subjects that have MCI. Yeah, I've seen the data. And the other thing is we also try to combine it with some other neurodegenerative diseases. We actually have the whole genome data for progressive supranuclear palsy, one of the largest data sets in it because it's such a rare disease. And we have, when we're talking in terms of longitudinal follow-up, you look at what's available for the phenotypes. Yes, some of these do have longitudinal follow-up, depending on what study you're looking at. The biggest one is, of course, the uniform data set for all the Alzheimer's centers, including Michigan. So they have different, different studies will have slightly different, interval for follow-up. I think it's not exactly uniform. It's not a clinical trial, but all these data are available. If you want to look at these, deeper phenotypes, you can go to NAC. You can apply for these data, and you can directly link with our genetic data. It's the same IDs, and then you can do the analysis. All these are there. Yes, Hank.
Thank you for the great talk, and I do encourage everyone to go to the NAC as well as all the tremendous genetic sites you're talking about. A key question right now, we learned so much biology from the genetics, is why some people progress rapidly and some people progress more slowly. And I'm sure genetics is a big part of that.
Yes.
I guess my question for you is, I know that the NAC data within the ADSP is longitudinal.
Yeah.
We have up to 10 years in some of those individuals. What other components of the ADSP 57,000 genomes do you have longitudinal data? Because I think it'd be great for someone to mine all that data for, let's say, the top quintile for fast progressors and the lowest quintile for slow progressors, and look for genetic differences.
Well, I can tell you some of the cohorts from the phenotype PSC, because what PSC does is it will look at some of the cohorts and add it to the bigger collection using a statistical model. So many of these, like A4 is a clinical trial, so it has a longitudinal follow-up. X has been doing longitudinal follow-up for a long time. It's University of Michigan, Washington, and Kaiser. ADNI certainly is longitudinal. IFIGA, I think most of them are longitudinal. Yeah. So like the clinicians, we have lots of records. Yes. But you need to take the data and decide how you're going to aggregate the longitudinal data into some aggregate variance so you can look at the rate of decline. And I think some of the challenges, that's why it's interesting these cognition are harmonized, because with different studies, you use different kind of instruments that have different sensitivity at different levels, right? The MMSE is going to be really bad at early diagnosis and so on. And yeah, it's a challenge. It's a challenge, but I think we have the resource to allow people to do it.
Dr. Wang, yeah, it's great talk. I have two questions. One question is about the data interoperability. So, for example, right, you have many databases, resources, like you even have the HRS, right, health and the retirement study from UMich. Actually, I contacted them before. So, like the HRS study, they usually surveys, questionnaires, and a lot of answers. So my one question is how you integrate this into your certain, or it's just like set aside, just its own independent work. So another question is about, I'm interested in also like occupation, how occupation or jobs can affect the outcome. So do you have the occupation record in all the data sets?
Well, we certainly don't have the occupation harmonizing. We don't have that. And I don't think all of the data sets will have occupation. And when you think about, if you want to ask the question about occupation for old people, it becomes really challenging, because that's, you're looking at what is their past working records, right? It becomes really complicated. For HR's data, we are the repository because we're NIA repository and they want to use our investment, so they put the genetic data into us. But what that also means, we have an opportunity to make sure that data and ADSP data are using the same genome build, have the same variant. So the analogy, it's not exactly that they're asking the same question. HRS has some cognitive decline data. It's I forgot what the name is called, but they have an instrument for looking at cognitive decline. It's not as comprehensive as your typical dementia study. Yeah. So and the other thing is, I don't think they have the diagnosis for dementia in their data. There are many aging study follow up like longitudinal study for health of retired people. Yeah. But there's still some things that are common. They actually, what they did is actually they have a sub-study in there that actually try to come up with a model that can impute into their dementia study. It's called ADAM study that give you some kind of score that tells you if the person has dementia or not. But it cannot replace the true diagnosis. Yeah.
I just had a couple of questions. So one is, I know in the beginning you had like over 75, maybe even hundreds of loci, right,
Yes.
that have been associated, and you showed the pathways. Have people started looking at protein complexes to see if some of these are actually, aside from just pathways functionally, because I actually say this because way back when in my postdoc, I was kind of doing this kind of thing, and what we found is in model organism and C. elegans genetics,
yes,
when you have a similar phenotype, oftentimes you get genetic mutations in the same protein complex. So that kind of like points, I think, more specifically even at causality. I was just curious if people looked at that yet. So there are big protein interaction databases. I mean, they're not perfect, but in terms of quality.
Oh, I think people must have done that kind of like pathway network interaction analysis.
Yeah.
Pretty common in those things. There are some studies that's kind of related. Someone in ADSP developed this tool that actually tried to look at the interactions between like SNPs by looking at their proximity in the 3D structure and see how that can detect interactions, improve statistical power. So that's one approach. But looking at the protein complex for Alzheimer's, certainly there's a history for this. For the early onset, when we're looking at it, they're looking at like, we know many of the variants, many of the genes in the association findings are part of the gamma secretase complex, right? So presenting 1, presenting 2, we know they actually cleave the APP. And of course, gamma secretase does like lots of other things, in particular the Notch pathway. So I think people wonder what these overlaps are, but I don't think we look at it deep enough. Yes, but that's a really important idea. Yes.
And the other question I had was, and I think it's along what lines what Margit said, but when you have these loci that have been identified, and with all these numbers, I mean, maybe people must be doing this, but like, how about those participants who had the disease risk allele but didn't get sick?
Oh, you're talking about resilient subjects. Absolutely. Well, like there's some. The first is you need to find data that has the kind of you have a way to measure neuropathology, right? So there are some studies that collect extensive autopsy data. The most famous one is the Rose map data, Rose and the MS study. This is a religious order. These are the Rush University. They have this like lifelong longitudinal follow-up and then at the end they donate the brain. So they did find these cases where you have people with lots of neuropathology like plaques in the brain. Their memory, their cognitive is perfectly fine, and we want to look at genetics of that, but it doesn't happen that often. There's a work group in ADSP looking at protective variants. I don't think they found any like low-hanging fruit at this moment. I
guess the numbers is the issue. Numbers,
I think numbers are absolutely. Actually, at the beginning, and Dr. Benke is here, he can comment on it. When we started ADSP analysis, some of the things that we're thinking was let's sequence ApoE, E4, homogeneous people and see if there's like proteins. But it turns out that when you have E4, the chance, like pretty much almost all of them get Alzheimer's. So we don't have enough sample. But that's the kind of study we had at the beginning. I hope at some point we'll have enough statistical power that we can look at protective variants. Yes.
So I have a one more general question.
Yeah.
So right now, I think AI just everyday
Yes.
life thing. So I was wondering, you mentioned that the AI subgroup in the ADSP as well as some API stuff. I wonder what's your vision about the AI and the large data set on Alzheimer's disease associated studies?
Yes, there are actually many studies in AI. They're really using very creative methods. For example, someone is looking at a model that can look at the evolutionary selection pressure at the different loci and use that to do gene discovery. And they're, of course, using AI methods to build predictors by integrating omics data, image data. There's this big concern called AI480 that does that. And one other thing we're trying to work out is develop some kind of benchmark data that has very well trained. So you can try different kind of methods and compare them. So there are all these different kinds that people can do. So I think what we want from the repository perspective is you have this data ready so people can try their new methods and have a way to compare. And then we want to make it as easy as possible so they have the data that they don't need to spend a lot of time cleaning things up, and they have complete metadata that allow AI to reason automatically, right? These are all the things we want to do. But the other thing I'm also wondering is some of the things we do in terms of building the infrastructure, harmonizing the data, and so on. We want to see how AI can help us with that as well. I think there's a lot of creative things we can do. We're talking about, Dr. and I talk about using ontology to do some of these things. Yeah, it's really exciting. It's just with all these ideas, how much time do we have to build these? Yes. I don't know if that answers your question.
Yeah, thank you. Any more questions? All right, with that, let's thank Dr. Wang again.
Thank you so much.